Yeah, I'm a PhD student in the Biological Sciences department, most you know, and I'm going to talk about how stem cell differentiation shapes the evolution of reproducible morphogenesis. So this is a computational modeling study in the area of evolutionary developmental biology. So I want to start off with talking about what morphogenesis is. So morphogenesis is the generation of the organism shape from a relatively homogeneous starting material, which is usually a fertilized zygote. And morphogenesis creates all the complex structures that make up our body, like our limbs, our organs, our tissues. And really an essential feature of morphogenesis is that it's reproduced from generation to generation. So when you have an offspring, their limbs, their tissues, and their organs are reproduced in the same locations relative to each other, just like yours. And understanding how this complex process of morphogenesis can be reproducible has intrigued the minds of people since Pythagoras and Aristotle. So I just want to reiterate that reproducibility is really important for morphogenesis because it allows an organism to inherit their ancestors' phenotype. And as we all know, inheritance is essential to biology. So before we talk about reproducibility of morphogenesis in more detail, I just want to speak about how morphogenesis occurs. So at a very broad level, morphogenesis involves the precise coordination of molecular level processes like gene regulation and biochemical gradients in combination with cellular level processes like cell division, migration, apoptosis, and differentiation. And so what we really want to know is how these processes are reproducible, or how these processes give rise to reproducibility. And in order for these processes to be reproducible, they have to be robust to the inherent randomness and noise that occurs in biological systems. However, it's been hypothesized that this reproducible morphogenesis sort of comes for free when you have the precise interplay of those many molecular and cellular processes that make up multicellular development. So when you have all these complex interacting things working together, reproducibility sort of naturally comes about from morphogenesis. So what I've been asking with my research is whether morphogenesis is an inherently reproducible process. So if you can have complex morphogenesis, it is automatically reproducible, or whether there are specific mechanisms needed to make morphogenesis reproducible. So the research I'm going to go over today is outlined as follows. What I did in a computational model is evolve the morphogenesis of complex shapes. And by evolving the morphogenesis of complex shapes, it's naturally going to require the precise coordination of those molecular and cellular level processes that I mentioned before. And then with the morphogenesis of those complex shapes, I can test whether they are reproducible or not. And I'll spoil you now, the central result is that most of the time, complex morphogenesis is not reproducible. However, the minority of organisms that do have reproducible morphologies have a single feature in common, and that is the stem cell system. And the stem cell system involves a population of stem cells that self-renew to maintain their population over time, and at the same time, irreversibly differentiate into differentiated cells. So let's go over my model for morphogenesis. So there are 4 key features to the model, and these key features are just the essential features of multicellular development, which are gene regulation, cell adhesion, cell-cell communication, and cell locomotion. And these features are implemented in a stochastic model called the cellular Potts model, which reflects the stochasticity, which is inherent in the kinetics and geometry of cells. So I'm going to play a video now of how the model works. So this here is an organism, and it's surrounded by some external medium. And you'll see each of these enclosed regions here is a cell, and the color of the cell is determined by its state. So let's explain what's going on there. So each cell in an organism is made up of a neighborhood of pixels on a 250 by 250 grid. So this green enclosed region here is one cell, and the cell is going to be surrounded by some medium, and at the same time, a cell was going to be next to 2 other cells. And the cells grow, shrink, and divide and move around. Grow, shrink, and move around by these stochastic pixel copy attempts that occur at the boundary between cells. So a pixel can copy itself into a neighboring cell, or between the cell and the medium. So a pixel can copy itself from the medium into a cell or otherwise. And that's the central dynamic of the model, just these pixel copies that occur. And you'll see that these different cells have different colors, and that color is determined by the state of the cell. And what the state is the state is determined by what proteins the cell is expressing. So essentially it's protein expression pattern. So each cell has a genome, and the genome is going to be the same for all cells in an organism. And the genome encodes these transcription factors, these blue nodes here, this triangle and this square, which activate and inhibit the expression of each other. So they activate and inhibit the expression of other genes, which is this wiring here, this inhibition and activation. And this wiring is called a gene regulatory network, and that's what we're going to evolve. So we're going to evolve this wiring here. And you'll see that a subset of these transcription factors, specifically this square and this triangle, are morphogens. And they are a special subset of transcription factors because they can diffuse in space and decay over time. And the purpose of those morphogens is to allow for communication between cells. So because they can diffuse in space, they can set up gradients, concentration gradients. And because cells can be exposed to different concentrations of morphogens in space, they can respond by expressing different proteins. So we see here these concentric circles represent the concentration of morphogen from high to low. And depending on what concentration of morphogen a cell is exposed to, that can cause it to express different proteins. And these morphogens are, yes, they are very essential for development in both animals and plants. So the next thing I model is cell adhesion. So the transcription factors can activate and inhibit the expression of these adhesion proteins that I've circled here in these red circles. And these adhesion proteins are going to facilitate binding to other cells. And the way that binding works is via a lock and key mechanism. So if one cell expresses a lock and the corresponding cell, and a neighboring cell expresses the corresponding key, the lock and key will bind together and increase the strength of adhesion between those two cells. So I'll explain how that works dynamically. So let's say you have some green cell here, which is bound to this orange cell. So the green cell expresses some key and the orange cell expresses some lock. And let's say this green cell changes state, changes color, and it no longer expresses this key protein. And that means its adhesion strength to this orange cell is going to change. And then through those pixel copy attempts, which is the main dynamic of the model, the cell can move away because it no longer is strongly bound to that cell. So I also model cell growth and division. So because cells will move to maximize their adhesion with surrounding cells, it can cause them to stretch. And when a cell is stretched, it can increase in size, and the cell will just divide in two if it reaches 100 pixels in size. So now I want to talk about how I initialize an organism at the beginning of its development. So an organism is initialized as a ball of 64 cells. And you'll see that the color of these cells are different. And the reason they are different is because I initialize two transcription factors to different concentrations depending on their location in the initial organism. So these numbers here just represent the initial concentration of those two transcription factors. And the reason I model this asymmetric distribution is because it's really important for axial symmetry breaking. So asymmetric distribution of proteins will very commonly give rise to the top and bottom and front and back asymmetry that we see in extant organisms. Given this model that I've spoken about, if I create some randomly wired gene regulatory network, so this wiring of this gene regulatory network, I just generate a random one and simulate the development of an organism, which I'm going to play a video of now, you'll see that the organism doesn't actually do anything. It just remains as a ball of cells, and all the cells are the same color. And the reason they're all the same color is because they're all expressing the same proteins. So there are actually no interesting dynamics going on there. So what I do is I computationally evolve the morphogenesis of complex shapes, which is going to necessarily require the precise coordination of all these parts of the model. So how that works is I have a population of organisms, and each organism will undergo development on a separate grid for a fixed period of developmental time. And once they finish development, what I'll do is I'll select for organisms with the most complex morphology. And the way I select for complex morphology is by how much the organism deviates from a circle and how much inward folding there is. So we see with this organism here, it has inward folding, and it's clearly not a circle. And the same for this one down the bottom here, but the one in the middle, it just remains as a circle through development. So that one in the middle will die and the other two will reproduce. And during reproduction, mutations can occur through that gene regulatory network. So an example mutation is shown here. Let's say we have some transcription factor X that inhibits the expression of gene Y that can mutate to activation during reproduction. And then the next generation will develop, and I repeat that process over and over again. So I ran simulations of this model for at least 2,500 generations, so lots of simulations, and evolved complex morphogenesis and asked whether that morphogenesis was inherently reproducible, so it's just automatically reproducible, or whether there are specific mechanisms needed to make it reproducible. So here's some example organisms that I evolved from my simulations. So 89 out of the 125 that I ran resulted in organisms with complex morphology. And what I'm showing here is the evolutionary trajectory of one simulation. So on the y-axis, we have the average fitness in the population. And on the x-axis, we have the number of evolutionary generations that the simulation's gone through. And these organisms are taken from successive generations through this evolutionary process. So at the first generation, the organism's just a simple ball of cells. If I take a random organism from that population, and then over evolution, we see the evolution of some complex morphology. And this organism here is taken from the final population. And I'll consider this like the evolved organism and use that for further analysis to test whether it's reproducible or not. So I'll show you what the development of that organism looks like. So it starts as a ball of cells. And then as we see, as it develops, cells down the bottom here are going to proliferate and make some sort of cup shape. So we see we get inward folding here and here and make a nice stalk in the middle. And here's another example of an evolutionary simulation where complex morphogenesis evolved. And the way I can test for reproducibility is by replaying the development of an organism over and over again. So because the model is stochastic, it needs like a numerical seed to initialize the development. So if I run the development of an organism with a different seed, there's going to be a lot of noise and randomness that's going to change how that organism develops. However, when I replay the development of this first organism that I showed you, it makes approximately the same shape each time it develops. So I'm showing you here the organism at the beginning and at the end of its development. So you see at the end of development, it's got approximately the same shape. In contrast, when I replay the development of this organism down the bottom, so these are 4 replays, it makes quite a different shape every time. So what I did is I came up with a rigorous measure to determine complexity by replaying the development of an organism 60 times and measured how similar the morphologies were. And this measurement is invariant to rotation, reflection, and translation. I won't go over that in detail though. And the end result is this distribution of reproducibility scores. And what I found is that there is a large distribution in reproducibility scores, and that distribution was actually sort of bimodal. So some organisms are actually quite highly reproducible, as I'm showing you 4 examples of here. These 4 examples at the top are highly reproducible, but most organisms are actually not reproducible. So these 4 organisms down here are examples of organisms that aren't really that reproducible. So to answer my initial question, clearly no, morphogenesis is not an inherently reproducible process. So the next thing I did was try and identify what mechanism could be causing these organisms to undergo reproducible morphogenesis. And what I found was that all the organisms that underwent reproducible morphogenesis had one characteristic in common. They had populations of proliferating cells, which are these cells down the bottom here, if you remember the development of this organism that I just played, that differentiate during development. And this process resembles stem cell systems, which are essential for development in both animals and plants. So what I did to rigorously test for the presence of stem cell system in each evolved organism was test for the two key characteristics of a stem cell system, which are irreversible differentiation from a stem cell to a differentiated cell, and self-renewal of stem cells. So they're actually dividing and maintaining their population. So this organism here, which does have a stem cell system, I won't go over the tests in detail, but the result from testing for irreversible differentiation is this here, which is called a state space. And what this state space shows is how cells in certain states are able to transition to other states. So we see here, if a cell is in this light blue state, once it transitions to this purple state, there's no longer a way for it to transition back. So the differentiation is actually irreversible. So when that occurs, I designate these upstream states as stem states and these downstream states as differentiated states, and test whether these stem states are proliferating, so whether they have self-renewal. And in the case of this organism here, if you remember the video, you saw that these cells down the bottom here, these stem cells were proliferating. So this organism has a stem cell system. In contrast, here I will play you the development of an organism that does not have a stem cell system. So we see, if we look at the state space from this organism here, and we look at how cell states transition to each other, any cell state can transition to any other cell state. So there's a pathway between all cell states. So there's clearly no irreversible differentiation, and it does not have a stem cell system. So if I take those reproducibility scores and separate them by the presence of stem cell systems, what I find is a clear distinction between the reproducibility scores of organisms that do have stem cell systems on the right from organisms that don't on the left. So organisms with stem cell systems are clearly more reproducible, and that's where the distribution seems to be coming from. So the next thing I asked was, what is the mechanism explaining this difference in reproducibility? So why are stem cell systems more reproducible? And the answer I'm going to explain to you relates to two things. The first is the number of symmetry breaking events that occurs during development. And the second is the degree of anisotropy that arises from each of these symmetry breaking events. So symmetry breaking refers to when uniformity of cell properties is lost. So let's say we have some ball of green cells, where the green just means like their cell state, and half the cells change to an orange state. So that's a symmetry breaking event because it's caused the cells to lose some uniformity. Now, anisotropy refers to the directionality in the cell locomotion that occurs after symmetry breaking. So let's say these orange cells move and grow in a certain direction, whereas the green cells don't. There's clearly anisotropy to how these orange cells are moving, and as opposed to isotropy, which means they're all moving in the same direction. So these orange cells are clearly moving with a certain direction in this schematic I'm showing you here. And that anisotropy is something we can quantify. So my hypothesis, what I'm going to explain to you, is that organisms with stem cell systems can undergo complex morphogenesis from the anisotropy produced from a single symmetry breaking event. In contrast, organisms without stem cell systems need to break symmetry multiple times to achieve the same degree of complex morphogenesis. And symmetry breaking is prone to noise, and because it's prone to noise, the morphologies of these organisms without stem cell systems is less reproducible. So let's explain that in a little bit more detail. So what I'm showing you here is the development of an organism which has a stem cell system. Now, this organism is quite nice because it has two types of stem cells, the type 1 stem cell, which are these green ones, and the type 2 stem cell, which are these orange ones. And you'll see that these type 1 and type 2 stem cells both differentiate into the same blue type of cell. So those are the three cell types of this organism. So the first thing I did was ask how this organism breaks symmetry, and it breaks symmetry at the beginning of development by that asymmetric distribution of transcription factors, which specifies the type 1 and type 2 stem cells. So that's the initial configuration of cell types. And the next thing I asked was, okay, so given this initial symmetry breaking event, how does anisotropy occur? And what I did is I summed up all the velocities of type 1 stem cells, differentiated cells, and type 2 stem cells independently and tested how the cells are moving through development. And what I found is that these type 1 stem cells clearly have directionality to the way that they move. These type 2 stem cells also clearly have directionality to the way that they move. And these differentiated cells don't actually move at all. And what I found is, the next thing I asked is where that anisotropic growth comes from. So why are they growing with directionality? And what I found was it actually relates to cell differentiation. So the way cell differentiation occurs is that these blue differentiated cells actually secrete a morphogen signal which tells the cells around them to differentiate. So when a type 1 or type 2 stem cell comes into close contact with already differentiated cells, they will also begin to differentiate. And because of that, there is directionality to differentiation. So they'll always differentiate in the same way. And because they're differentiating in the same direction, they also know where to grow because they can grow in the direction that they're not differentiating. So this organism will grow away from the differentiated cells and differentiate towards the differentiated cells. So what this means is the communication between cells is clearly very important for its development. So the next thing I asked was, okay, so what if we take away this communication? How do the cells develop? So what I did is I made balls of cells of this type 1 stem cell, this differentiated cell, and this type 2 stem cells. So there's only one cell type in each ball, and develop and see what happened. And what I found is that these type 1 stem cells just all grow isotropically, so they just grow in all directions. These differentiated cells don't grow at all, and these type 2 stem cells don't really grow either. And importantly, they just remain as balls of cells through development, so they don't actually undergo complex morphogenesis by themselves. And what this means is that the interactions between cells, the communication, the cell-cell communication is essential for complex morphogenesis to occur. So if I measure the anisotropy that occurs in these individual balls of cells, it's clearly not anisotropic, it's just isotropic growth or not growing at all. Whereas in the actual development of this organism, it can undergo anisotropic growth because of the cell-cell communication. So what that means is because the initial configuration is approximately the same each time it develops, the communication between cells is going to play out in the same way. And what that means is the same morphology is going to present itself each time we replay the development. Even if there's quite a bit of noise happening. So how can we sort of test this hypothesis further? So what I'm going to do now is introduce an artificial symmetry breaking event. So I'm going to take some differentiated cells on the right side of this organism and revert them back into the type 2 orange stem cells and see and test whether the behavior of those reverted stem cells is predictable. So whether they can grow and differentiate in a similar predictable pattern. So let's play a video of that. So right here, you see I revert these differentiated cells back into orange stem cells. And if we watch what happens, they actually create a structure that resembles the nascent structure down the bottom here. They grow away from the organism and differentiate towards the organism. So there's clearly a predictable pattern of growth and differentiation that's occurring. Now, if I compare that to organisms without stem cell systems, what I find, as I explained before, they're not reproducible. And the reason they're not reproducible is because although they can use the asymmetric distribution of transcription factors to break symmetry at the beginning of development, they will also break symmetry in random locations as development is played, and that's promoted by being undifferentiated because they don't have irreversible differentiation. So if we have a look at the development of this organism, we'll see that these sort of these growth zones, or these sort of buds, tend to grow out from the organism at sort of different regions as development goes on. So it's actually breaking symmetry and developing in different locations. And doing that actually increases the complexity of the shape. So see, because it's growing these buds out here, it can increase the amount of inward folding and complexity of the shape there. But those symmetry breaking events that are occurring are not reproducible and highly prone to noise. Not reproducible because they're highly prone to noise. So when I replay the development, it'll break symmetry in different locations every time. So this led me to hypothesize that there was a trade-off occurring. So an organism can break symmetry more and increase its morphological complexity because it can grow in lots of different ways. However, as it increases its complexity, it becomes less reproducible. In contrast, stem cell systems might be a way to circumvent this trade-off because they can maintain reproducibility while being morphologically complex. So the way I tested for that is look at the evolution of both fitness, which is like morphological complexity, and reproducibility. So what I'm showing you here is two simulations, one where a stem cell system evolved and one where a stem cell system did not, in orange. And we see that their fitness or morphological complexity reaches approximately the same score at the end of the simulation. So they're under my metric for morphological complexity, they reach about the same score. However, if we look at reproducibility, at the beginning of simulations, both simulations have organisms with reproducible morphologies, and that's because the organisms have trivial reproducible morphogenesis. So if we look at the organisms at the beginning of simulations at generation 100, they're very simple shapes. And if you have a simple shape, you can make that simple shape every time, right? So you're obviously going to be reproducible. However, as complex morphogenesis evolves, the decline in reproducibility is much stronger when there is not a stem cell system compared to when there is a stem cell system. So what I did now is I repeated these measurements for many simulations by measuring the reproducibility and the morphological complexity of many organisms throughout the simulation. So organisms at the beginning, in the middle, and at the end, and measured the trade-off between reproducibility and fitness, which is morphological complexity. And what I found is that this trade-off between reproducibility and fitness is much weaker in simulations where stem cell systems evolved compared to simulations where they did not. So what that means is stem cell systems is allowing organisms to circumvent the trade-off between complexity and reproducibility. And the reason that is because they can make a more complex structure from just one symmetry breaking event compared to organisms that do not have stem cell systems. What time is it? 58. Okay, I'll stop there. Thanks for listening.